Thanks very much, Marcus, for this kind introduction, and also from my side, a very warm welcome to all of you. Thanks for your interest in this webinar, which is a grand journey today from 10 to the power of 16 opportunities, so many compounds, down to single compound, even atom precise visualization of delta G's in the binding site. So bear with me while I explain how we go from here to there. We do that using the workflow system KNIME as a vehicle. KNIME, as you all may know, is a general purpose environment that allows you to connect certain compute services with each other and construct this way a complete workflow. For example, like here, from reading a molecule to a 3D visualizer, and many more examples of that sort will follow. Why did we choose KNIME, and what can BioSolve IT do for you in KNIME? You see here, well, a lot. If you install all the KNIME nodes, all the BioSolve IT nodes in KNIME, then you get this component tree, and obviously all our commercial tools are covered here. But I also want to mention that quite a few free tools are included here as well, notably a 3D coordinate generation, as well as the Naomi toolkit for file conversion and filtering. And beyond that, we also contributed back to the community with, for example, an improved table view that is part of the KNIME environment, and which we improved in a number of ways. So that is all our stuff in KNIME, and what it comprises is what we are good at, and that is providing very special technology. And I'm proud to say that we are superior there in many ways over the competition to solve certain scientific challenges. For example, dealing with virtual chemistry spaces. You can generate the Colibri, you can use the Colibri nodes, for example, to generate a space that is so big that you can't even enumerate it. And I'm going to show you in a few seconds. Second scientific challenge is, well, now when you have such a space, you want to search and find something interesting actually in the space. And third for today is docking and scoring, both well-known problems in the virtual screening world, where you want to narrow down from lots and lots of opportunities down to the most promising candidates. So these are 3 examples of technologies from our side to solve scientific challenges. And solving those scientific challenges is something that we are particularly good at. The question now is, what is KNIME good at? Well, it is good at connecting the dots, the individual components for practical usage. So you see here some BioSolve IT nodes for library generation, searching, a docking node, and just as the name suggests, workflow systems combines them or allows you to combine them to a workflow to turn it into something really practical. practical use. It comes with the advantage of just one interface for the user to learn, so a very steep and quick learning curve, instead of this tool, this interface, next tool, next interface, and so on and so far, which would be a lot of work. It comes with convenience processing options. For example, you have multiple CPUs on your computer. You want to use them all. Well, just surround some nodes with the parallel chunk loop nodes, and there you go. It runs now using all your CPUs. So that is something that KNIME is particularly good at and strong at. And last but not least, aside of connecting our dots for generation, searching, validation, and visualization, as a user you have the choice to mix and match the best of many worlds. So we don't even claim to cover all aspects of this drug discovery game equally well. We don't have functionality, for example, to build a homology model. Well, that can be something that somebody else has solved, and if that somebody else provides 9 nodes as well, you can put these things together and you still have just this one interface. So you can pick the cherries, so to speak, from many baskets and connect them here to make something of practical usage. So let's start with generating a very big chemistry space. Well, you determine whether it's big and how big it gets. The tool behind this is called Colibri. You find it, of course, on our webpage. So here's a little bit of a motivation of this chemical space exploration toolkit. Imagine you could find anything in a huge chemical space. And by that I mean, if a molecule is in there, as our space consists of molecules, we want to be able and want to be sure to find it, not just by chance, but be sure. Secondly, from one place we want to jump to something distant yet alike. You know the usual dilemma. If you use any search method, you usually search by similarity. But then again, if it is just mere analogs of the compound of your starting point, this may be of less interest. So you want to have a search method that gives you something more interesting, similar, but not the same or essentially the same. And I'm showing you what I mean by this. And third, altogether, you want to have something interesting, but it must be real, like in a real space, meaning that you don't want to see just de novo molecules, but you want molecules that you can actually make afterwards in the lab. Okay, and all of this together, finding everything, jumping to something distant yet alike, translates to scaffold hopping, and interesting and synthetically accessible results. That's our motivating factors behind our approach to chemical space exploration. How do we do that? Well, it starts with generating and then comes searching. So instead of just collecting molecules and enumerating molecules, taking vendor catalogs, in-house compounds, you name it, instead of that, we collect reaction protocols. Actually, mostly protocols that are not just mere transformations, but reactions that connect reagents and building blocks to build larger molecular entities. And I'm not just talking about one or two or a handful of reactions. I'm talking about in the hundreds of reaction protocols. And you know that if you assemble pieces in a combinatorial fashion, then easily the space can get very big. So if you have, for example, some sort of coupling reaction and have 1,000 reagents of type A and 1,000 reagents of type B, and they are coupled, then you end up already with 1 million. And of course, this trick applied 100 times leads to a real big chemistry space. It contains billions of compounds easily. Okay, second is searching. There we take a particular similarity method called feature trees. It starts with a query molecule that is your compound of interest, say, and it provides you something similar, or as I said in the outset, distant yet alike. Yeah, you can see the similarity or partial similarity, and these particular molecules, well, I took them because these are documented to bind to the same target as the query. The details are not significant here, but that is exactly what we want to achieve. Find something distant yet alike, and the target tells us what should be alike. The added value from this is we still know that this compound comes from reaction X, Y, or Z, and we even know the composition of reagents that undergo a reaction here to form this product molecule. So we have a recipe associated with every virtual product. So generating the space and then searching in the space. I start with the end because I think the searching ultimately determines, or the ability to search ultimately determines the value of the method altogether. So let's start with, first one more slide about the space. We compiled a literature space, so that's literature reactions, more than 120 of them. And here you just see some examples of ring closures, substitutions, additions, multi-component reactions. So that is from taken from the literature, and as an input, we take vendor catalogs, so compounds or reagents that can actually be bought, so that you can have a real access to the molecule. And we turn this into a literature space. We call it knowledge space, and that comprises these billions of virtual products. So if you are a little bit afraid of part number 1, which was the generation of the space, we also provide a literature space. But I talk about the generation of the space again a little bit later. How does this connect again to KNIME as our workhorse here to connect the dots? Well, you have a number of options to import molecules from files, from databases, you name it. You have a number of filtering options, and we provide some of them too. So it's easy to implement, for example, rule of three, so fragment-like filter. Also KNIME easily allows you to get rid of unwanted substructures, stripping of salts, so that you can process the molecules that are in those databases. So that's the input stage, and all of that can, of course, be configured by the user. So very flexible. The second step is generating working with the reaction protocols. So you will have a drawing program likely, or a reaction reader on the one hand side, and then that filtered reagent set on the other hand side. And that goes into a so-called enumerator node. I show you a picture here of a reaction. It is one of these reactions in the KnowledgeSpace that I showed to you earlier. It's really just merely the reaction diagram of reagent 1, 2, and 3, and the new product core. So into the node, the enumerator node, goes this reaction description and the set of reagents, and that will compile a space just consisting of the molecules of this one reaction. You can enumerate it, so you can see what compounds are in them. You can use those enumerated product molecules, of course. But really interesting becomes the whole thing, of course, if you put them all together, and we have a specific tool, the fragment space merger node, that allows you to compile really from all reactions a single space and then write it out to file for later usage. So that is how the fragment space generation is wrapped in KNIME. And we provide this whole workflow so that you can actually do this yourself. You can take our reactions, but you can also define new reactions by drawing these reaction diagrams and feeding them into the space. And what you take and what you leave out is, of course, up to you. So all very convenient, and we have full control and flexibility over the process. Okay, now I come to the searching. Now that we have a space, either pre-compiled, so the one that we deliver, quote-unquote knowledge space, or one that you compile yourself. Now it comes to searching. Feature trees is the name of the method that we use to search in such a space. And the similarity, of course, starts with its descriptor. And I show you on a picture now how the two descriptors for these two molecules, by the way, happen to be, again, active against the same target, how the descriptor looks like. So this is overlaid now with the molecular skeleton the feature tree descriptor. So you see, it is a topological graph. It has nodes and connections in between. It's a tree structure, so no circuits in it. Instead, the circles are absorbed here into single nodes, and bicyclic or more complex ring systems are decomposed also in a fashion so that we can work with a tree topology. That is very important. Okay, and aside of the connections, you see that the nodes have colors, and the colors are just a symbol for they have properties, yeah, associated with them. But you see also one other thing, not every atom has a single node, but sometimes we collapse functional groups into nodes. And this gives us what we usually call a one level of abstraction from the exact molecule topology. And that is key for its ability to find us some of these remote similarities, as you will see later. Okay, these are the descriptors for the two molecules. Now comes the second important ingredient to a similarity method. First, the descriptor, second, the mapping method. So now we have an algorithm that maps the nodes of one tree onto the nodes of the other tree. And that leads to two nodes to be compared, and they get a local similarity. Yeah? So for example, this red one on this red one, highly similar. Yeah? This turquoise one onto this turquoise one, both representing an aromatic ring, also highly similar. Other nodes maybe not so similar. So the overall similarity is determined by the average of the local similarities, yeah, the average of these node mappings. And from all the possible mappings, the algorithm finds the one with the highest overall value. So we get the maximum similarity of 0.81 of these two molecules. And the added benefit of this way of calculating the similarity is we have the mapping, so we can use it. We can, for example, in 2D, use it for a color-coded depiction and see on the molecules what the algorithm maps onto what on the other molecule. For example, here a ring onto an open chain in the other molecule. And we can even take it further and do, on the basis of the mapping of the different substructures on these molecules, a 3D alignment. So that's very valuable information, of course, aside of the mere similarity. What kinds of results do you get? Well, the method from the outset was aiming at scaffold hopping ability. These rough descriptions of the molecules allow us to detect remote similarities, and that is the prerequisite of finding scaffold hops. And here you can nicely see compound number 1. It's a literature study done many years ago, actually. The query molecule and 2, 3, and 4 pictures of the top hits found with the feature tree method, all active against the same target. And you see here how gradually the actual structural similarity of the molecule decreases. It actually decreases quite rapidly. So we find, even among the very top ranks, a molecule like this with a remote similarity that is active against the same target. But these black lines here actually indicate all active molecules from this D4 antagonist subset in the database. And as you see by the increasing number of black lines to the top here, the method also gives you an enrichment. So it is not only by chance that this molecule is high ranking, the method enriches the number of active molecules if rank ordered by similarity in a data set. So it can do what we promised. It can do scaffold hopping. It is also orthogonal to other descriptors. This is a very busy slide. I apologize for it. As we said previously, you get all the materials so you can look at it in more detail afterwards and later. All I want to point out here on this slide is that we did a huge comparison of different similarity methods compared among each other and also with feature trees. And the key thing here in this study was to identify which of the compounds were exclusively found by just one particular method. And feature tree sticks out here. 43% of the active molecules were found exclusively by feature trees and none of the other methods. And this number is across different data sets fairly high. So it is orthogonal, as the headline says, and it adds value this way, even if you already use other similarity methods for searching. Okay, so we generated space, and I described to you how we search in the space using the feature tree similarity method. And the feature tree similarity method, actually with one extension module, allows not only the molecule-to-molecule comparison, but also the search of a query molecule in such a chemistry space. Now let's assume we have done the search and generated virtual hits in the space. And now we want to validate them. How is validation usually done in the computer? Well, it is done via docking and scoring. And our docking workhorse, even after 25 years, is still among the best you find on this planet. The FlexX method, since first published in 1996 in the Journal of Molecular Biology, has been cited more than 1,000 times, and many success stories among those publications. And we don't just utilize the old code and the old algorithm, but we improved it over these years. So the preparation of both the protein and the ligand regarding protonation and tautomer generation is all fully automated. You don't have to do anything at this stage and can trust the results. You can have replaceable waters. The metal ions are automatically coordinated and taken. This coordination geometry is taken into account during the docking process, and many more little but important improvements to the methods. Last but not least, the pharmacophore feature in FlexX is outstanding because it does not just filter the generated results to retain the ones that happen to meet the pharmacophore, but it directs actually the search method to find placements that are in accordance with the set out pharmacophore. But I don't have enough time today to go to all the details. The literature is out there, and you may have had the opportunity to work with the software already. I just want to mention, just recently we have completely refactored the code. It is now quote-unquote thread safe, so it runs on all the CPUs of your computer. It works on the standard operating systems, and recently we included a novel template docking, which works on the basis of an overlay of the maximum common substructure as an optional feature in the docking procedure. So that's docking. Next comes scoring. We have a very particular new development there. You may have heard about it also already. Again, I don't have too much time for the details. I just want to give you a glimpse at the method and highlight the particular characteristic of it. So the delta G estimate by the HYDE function is composed of just two parts, the dehydration part and the hydrogen bonding part, and it is a sum over all atoms, and that in itself, I come back to later, is very useful when it comes to visualization. Other than that, it compares the unbound to the bound state, and looking at the bound state here in detail, again pictorially, the dehydration is a calculation of the buried versus solvent accessible surface, and the hydrogen bonding is actually the count of the additionally found hydrogen bonds in the complex. And here are the formulas. They are both very simple, and most notably, there is no parameter weighting the two components one against the other. So the delta G of HYDE has never been calibrated with experimental binding affinities, and in that it is a unique scoring function and energy function that is actually applicable across a very broad range of energy estimation scenarios. Okay, so putting together the scoring function is really relatively simple, so it's super fast to calculate, only a few seconds. Other methods need hours and days even for it on much more hardware, while this runs on a single CPU. So it is really fit for interactive lead optimization. These atomic increments I mentioned already, we use them for the visualization. We map back the atom contributions and show the atomic contribution to the delta G in the binding site. Here you go. That essentially completes our journey from 10 to the power of 16 opportunities down to individual delta G contributions in the binding site. And since it was first published, now start to appear the first real life application studies where HYDE has been used and applied and successfully so. So it works in practice. Okay, so that was docking and scoring, and already leading towards the next point, which is the visualization, namely the visualization of the delta G estimate. We put this all together in a new graphical user interface called SeeSAR, and we usually call it light-white modeling because it has some of the key ingredients of a modeling platform, but it is not the all-in-one solution, of course, but rather than that, kept very simple and easy to use, and in that sense it is light-white. It allows for compound prioritization through the atom contributions to the delta G, very good for SAR exploration. I make a change on the molecule and I see on atomic detail what the effect may be. It actually helps this way also to guide the lead optimization process. What's the most promising next change I could make to optimize my molecule further? And in that, it supports this drug discovery cycle, which is understanding through seeing things, then interactively improving, and by this creating new ideas, and then the docking and scoring to validate, at least on a virtual level here. So that's the graphical interface to the scoring function. And putting it all together, yeah, we have here generating the space. I've shown you the nodes to do that. Searching the space is providing a query molecule and using the space as an input. Then the docking and scoring can be run in parallel in such a workflow. And last but not least, the results are fed into the SeeSAR node, which pops open as the viewer to look at the results and do the visual inspection. Bonus material, aside of the nodes, so the ingredients, we also put together more complex workflows for you in KNIME. We call them amazing workflows, and the one that I've showed to you is called Biosphere, which is exactly what I described, namely the generation and the search of a chemical space. Secondly, we have the MedChem wizard, which is good for compound analoging in the computer. And I have another slide on that, as well as another slide on STORM, which is library-based pocket exploration. So let's take a look at this bonus material. MedChem-Style Analoging, well, the idea behind is use any promising compound as a starting point. Second, apply the typical MedChem transformations. And here you see on the picture what we mean by that. Methylene shuffling, aryl ring analogs, isosteric replacements. We have a whole library of substructures that we use for these replacements, and so on and so forth. This is what chemists usually think of first in terms of what they could do with a molecule to make a more or less small modification to improve some property or affinity. So all of that, all of these MedChem transformations are applied, and then just as in the workflow that you have seen, we dock and score all the compounds and then open the results up in the SeeSAR viewer. So that is called the MedChem wizard. And last but not least, another more complex workflow that users relatively often ask for is a tool for a library-based pocket exploration. So let's assume you have a bound molecule in a situation like this. A core part of the molecule forming a nice hydrogen bond network, and then a more or less specific or unspecific side chain not contributing much to the affinity nor to the location of the molecule. What you may do then is take that substructure or core and do the same trick as we did in the chemistry space definition, namely use from a number of literature chemistry protocols some sort of coupling reaction, and this way form a whole library of molecules and then rapidly dock all the side chains, in this case, that can be attached and maybe fill this binding site a whole lot better. And here's just a picture of this sub-pocket exploration. You have some core part of the molecule deep down in the pocket here that is kept fixed, and then we have grown to this fragment all these side chains, with a lot of variability, of course, and you can then pick the ones that look best again in the computer. And again, the name of the workflow is Storm if you search for it on our webpage. So that brings me to the end of today's webinar, and of course we open the panel for any questions and discussions afterwards. But last but not least, I want to point already to our next webinar presentation. Dr. Anita Begert is talking about a project at MercaChem about CDK8 inhibitors, and this is on November 23rd. Again, look at our webpage to know about the upcoming webinars and register for it early. And with that, I'm closing my presentation, and